Welcome to my classroom. I am Nesli. In this video, I shall try to explain what Nernst potential is. I call it a theoretical potential because it cannot be measured in the body except for some rare cases, such as the case of a cell with only potassium leak channels, in which case the resting membrane potential of the cell is equal to the Nernst potential of potassium ion. So where and how are we going to use the Nernst potential? Basically, there are two types of forces acting on ions, pushing them into the cell or out of the cell through the ion channels. These are chemical and electrical forces. Chemical forces are produced by the concentration difference between the concentrations on the inner and outer sides of a cell. The unit is molarity. It is at the level of millimolars in human body. Electrical forces are the electrical potential differences between the inner and outer side of the cell membrane. The unit is volt, and the level in the human body is millivolts. When we are trying to understand the direction of ion movement through the ion channels, we look at the direction of the driving force. The driving force is the sum of chemical and electrical forces. We should be able to subtract or add them if we want to find the driving force. Unfortunately, their units are different. One is in millivolt, the other one is millimolar. Under these conditions, it's not possible to do the calculation. Fortunately, we have the Nernst potential, which helps us to express the chemical force in units of millivolt. Let's start by considering a theoretical situation in which there is only a chemical force but no electrical force. And let's start by an ion that contributes to the electrical activities in the cells of our body. I choose potassium for the first example. Let's draw our cell here. We all know that the level of potassium in the cells is high and the concentration or level of potassium outside the cells are low. So I try to represent this with the size of the letters. At this point, it may be useful to remind ourselves that we have two important conditions that have to be fulfilled if we want to pass an ion through the cell membrane. These are: one, a force that pushes it from inside to outside or outside to inside; two, an ion channel through which the ion can pass. So in this example here, we now have a force, which is the chemical force. We need to add the ion channels. So let me place the ion channels for you here. Now the force can be active through the cell membrane and ions can pass through it. At the very beginning, there is electroneutrality on the inside of the cell and outside of the cell, as well as on the inner surface of the cell membrane and on the outer surface of it, which means every positively charged ion is accompanied by a negatively charged ion. There's a good reason why I am talking about the cell and the cell membrane as if they are two separate entities. It is because all ion movements that we are going to use here to produce the Nernst potential and all ion movements that take place in the body to produce the electrical activities in the cells are taking place in a very small area that is restricted to the neighborhood of the cell membrane. So let us simplify our figure. Let's draw our cell membrane and concentrate on the positive and negative charges on either side of the membrane. Before we go on further, I have to remind you about a very important rule that you should keep in mind while listening to me in the rest of this video. This rule says that the number of ions, the number of ions that are going to move across the cell membrane is big enough to produce an electrical activity, but the number of ions is too small, I'm repeating it, to produce any change in the concentration of the ion. When the two conditions, the chemical force and the ion channels, are provided, potassium ions start to move across the membrane through the ion channels towards the outside of the membrane. For simplicity purposes, I have not drawn the ion channels in this figure, but I would like you to consider that they are still there. As potassium ions move outward through the cell membrane, through the channels, negatively charged ions may try to follow them. However, they cannot, because there are no ion channels present on the cell membrane for these anions. Under these conditions, negatively charged ions will start accumulating on the inner side of the cell membrane, while potassium ions accumulate on the outer side. This creates an electrical potential difference. The negative end of the electrical potential will be on the inner side of the cell membrane, and it will start pulling the positively charged potassium ions back into the cell. Under these conditions, we shall have two types of forces: a chemical force and an electrical force. The chemical force produced by the concentration difference will be represented by a black arrow, and the electrical force that is produced by the electrical potential difference will be represented by a red zigzag arrow from now on. Let the magnitudes of our arrows represent the magnitudes of each force, and let us follow the changes that take place in each of these forces as potassium continues to go out of the cell. Here is our force, chemical force, pushing potassium out of the cell, and at the very beginning, the electrical potential is very small compared to the chemical force. So, let's draw it like this. So our net force is still outward, and potassium will continue to move outward. The net force is also called the driving force. Let us represent the driving force by a green arrow. So the driving force will be the difference between the electrical and chemical forces, and now it is smaller than the chemical force itself, because some of the chemical force effect is eliminated by the electrical force, but it is still outward, and so potassium continues to move out of the cell. As potassium continues to move out of the cell, more and more ions will accumulate on the outer side, positive potassium ions, and negatively charged ions will keep accumulating on the inner side of the membrane. This is going to increase the magnitude of the electrical potential. So let us represent the electrical potential, this time by a longer arrow representing a bigger magnitude. Now at this point, I need to be very careful about what is happening to the chemical force magnitude. I need to remember that the number of ions that produce these electrical changes are not enough to change the chemical force or the concentration difference. So I must make sure to draw the size of the concentration force in the next step as big as the first one. So the steps will continue like this as more and more potassium moves out. And as you see, as more and more potassium moves out, there's no change in the concentration difference or the chemical force, but electrical force grows as the net force decreases. So in the next step, the chemical force will stay at the same magnitude. Electrical force, its magnitude is going to increase further, and the magnitude of the driving force shall decrease further. Potassium continues to move out of the cell. Eventually, electrical potential becomes equal to the chemical potential, at which point they cancel the effect of each other because they are in opposite directions. Here is our chemical force. Electrical force. And the net force, the net driving force, is going to be 0 at this point, at which there will be no net ion movement through the channels towards either side of the cell membrane. This point is an equilibrium point, and the electrical potential difference formed at this point is called an equilibrium potential or Nernst potential. Here we have discussed how the Nernst potential for potassium is formed. Let's go over the basic characteristics of the Nernst potential. 1, the Nernst potential is an electrical potential formed on 2 sides of the cell membrane. 2, it is produced by the concentration difference. Since it is produced by the concentration difference, we need to be careful and understand that it can never get to be bigger than the potential, than the chemical difference itself. So, the third characteristic is that the force of electrical origin, the Nernst potential, the force of it is always equal to the force of the chemical difference. Fourth is that this force is in opposite directions with the concentration difference, the chemical force that produces it. Nernst potential is a potential that is produced by a concentration difference. If there's no concentration difference, there will be no Nernst potential. And if you want to change the Nernst potential, you have to change the concentration difference. Now let us try to consider changing the concentrations of potassium on the inside of the cell and outside of the cell, intracellular and extracellular fluids, to see how this is going to change the Nernst potential of potassium. Here I have drawn 5 copies of the same cell. Same cell means that I will only change one condition in all of these 5 figures, and this condition is the concentration difference for potassium. For the cell at the topmost, it is going to stay as our original cell, so that we can remember the levels of potassium by looking at it. For the cells on this side, for downward, for these 4 cells, I'm going to change the concentration difference. There are 2 ways of changing the concentration difference. Either I can play with the inside concentration. So on the left side, I'm going to play with the inside concentration, or I can change the outside concentration. So on the right side, for you, I am going to change the concentration on the outside for potassium. So, how can we make, the next step is playing with the inside concentration. What can we do? What is going to happen if we decrease the concentration on the inside? The concentration difference is going to change, and if the concentration force is this big in the original figure, it is going to be smaller in the cell under the new conditions. So, if Nernst potential is the electrical potential that is equal to it by force but is on the opposite direction, what do you think is going to happen to the Nernst potential? Basically, we say that it becomes less negative. So, what does less negative mean? Let me open a parenthesis and explain it here. Whenever we are talking about a cell, the membrane potential, we always refer to the inside of the cell. So, for the original cell, the inside is negative. For the cell number 2, the inside becomes less negative, which means the membrane potential becomes less negative for us, because whenever we are talking about the membrane potential, we always refer to the value on the inside. So, by decreasing the inner potassium concentration, you decrease the concentration difference, and so the Nernst potential becomes less negative. What about the third example here? If you increase the inner, we are playing on this side, we are playing with the inside concentration. If you increase the concentration of potassium inside, the concentration force is going to be bigger. So the Nernst potential, which is equal to this force, is also going to be bigger. But saying bigger is not a very good way of expressing it. We better say it becomes more negative. So these are the examples of how we can change the Nernst potential by playing with the inside concentration. Let's move on to the outside concentrations. If I decrease the outside concentration, what is going to happen? The concentration difference is going to increase, And the Nernst potential is going to be more negative. While playing with the outside potential, if we increase the outside potassium concentration, then the Nernst potential and the concentration difference, both of them are going to be smaller. And in this case, we say that Nernst potential for potassium becomes less negative. So this was an example of how we can change the Nernst potential. It basically depends on the fact that Nernst potential itself is created by the concentration difference. The only way you can change it is by changing the concentration difference. Hope to meet again in another video in my classroom. Have a wonderful day.